All right, we can get started. Hello, everyone. My name is Ashley Sinks, and I'm the host for today's webinar. Today, we'll be discussing how to support a holistic safety program with microbial decontamination. Please note that this is being recorded, and we will send out the recording after the webinar is over. If you have any questions, please feel free to post them in the chat. We will definitely have time for a Q&A portion at the end. Today, we will be hearing from our two experts, Erin Mertz and Joy Herdt. Erin Mertz is our Director of Global Food Safety and Public Health here at Ecolab. She has over 19 years of experience and offers expertise in microbiology, regulated product development, and technical support for food processing environments. She holds a Bachelor of Science degree in microbiology and is certified in HACCP and a certified preventative control qualified individual. Joy Herdt is our senior staff scientist at Ecolab, and she brings over 29 years of experience in developing tailored solutions for the food and beverage, institutional, and pest elimination markets. Her focus lies in the development and registration of antimicrobial treatments for a safer, cleaner food supply chain. Joy is named inventor on 16 U.S. patents and has contributed to the book Microbial Safety of Fresh Produce. She also holds a Bachelor of Science degree from the University of Wisconsin. Today, we will be looking at three key areas. First, we'll be looking at microbial challenges, specifically taking a look at what's making headlines, Micro 101, and food safety and food processing. Next, we'll be looking at chlorine dioxide. And during this section, you'll hear an overview about chlorine dioxide and get more information on the why, when, where, and how it's used. And lastly, we'll be looking at what best practices you should keep in mind when leveraging the power of chlorine dioxide. And with that, I will hand it off to Joy and Erin.
All right. Thank you, Ashley. First, we're going to start with what's been making headlines. If the past few years have taught us anything, it's that this control of bacteria is not easy. Arguably, the most impactful outbreak in recent years was Coronabacter sakazakii in powdered infant formula and the subsequent formula shortage that followed. This highlighted issues and challenges not only in the processing environment, but also in our regulatory bodies that oversee the critical processes. This outbreak may have been a factor in the recently announced changes in the FDA and the creation of the Human Foods Program. Another big one is the recent outbreak of Listeria monocytogenes in boar's head liverwurst, and the resulting illnesses and deaths that could potentially initiate more change in how our food is regulated and inspected. This outbreak was also highlighted the importance of plant maintenance, sanitation, and the control of environmental pathogens. Others identified this year: salmonella in eggs, mangoes, cucumbers, fresh basil, the end of the 10-year outbreak of Listeria monocytogenes in soft Mexican cheeses, or I should say the identification of the problem food there, as well as Listeria monocytogenes in salad kits, and E. coli 0157H7 in onions. So let's take a step back and level set on what we're talking about today. When referring to microorganisms, specifically those related to foods, we're generally referring to the following groups. First, and arguably the most important when talking about our environmental food safety, environmental, not general food safety, is bacteria. Bacteria can cause both food safety and food spoilage issues if not controlled. Common examples of foodborne pathogens, Escherichia coli or E. coli, Salmonella species, Listeria monocytogenes, and some other ones. Spore-forming organisms like Bacillus species and Clostridium species are also bacteria. Next is yeast and mold. These organisms also cause food safety and spoilage issues. Mold examples are Penicillium and Aspergillus species, and common yeasts are Candida albicans and Saccharomyces cerevisiae. Viruses are predominantly a food safety issue, not necessarily a food spoilage issue. Common foodborne viruses are norovirus and hepatitis A. In the non-foodborne category, but important at this time of year or coming into this time of year in their importance, is influenza, coronavirus, and respiratory syncytial virus or RSV. Finally, parasites. These organisms are also predominantly a food safety issue, not a spoilage issue. Two examples include Cyclospora, which you hear about mostly in the summer months and can be imported from tropical areas on leafy greens, produce, other produce types, and Cryptosporidium, the culprit of a large waterborne disease outbreak in Milwaukee, Wisconsin in 1993. For those of you who are old enough to remember from this or are from this area, this was a big deal and one of the largest waterborne outbreaks to date. When it comes to microorganisms, not all are bad. We have the good, the bad, and the inert-ish. The good, we just mentioned some not so great examples of microorganisms, but not all are bad. Microorganisms can be beneficial to us and do great things, even save lives. An example of this is in the industrial insulin production. In a lot of cases, this is done using the bacteria E. coli and the yeast Saccharomyces cerevisiae. Of course, we cannot forget about all the delicious fermented foods and beverages. Bacteria, yeast, molds, they all have roles to play in fermentation in the fermented foods we love. And there are a lot of beneficial bacteria in your gut that help you digest food and outcompete bacteria that could make you sick. Then there's the bad. When you think of microorganisms, most people think of the bad, which is fair. But a lot of microorganisms can cause disease, and even those not thought of as pathogens can cause disease if the conditions are right. They're also very good at making food go bad and producing off flavors and smells. And finally, the inert. I've called them indifferent before if you guys have heard me speak, but this isn't a fair name for what these organisms are. They don't fall neatly into the helpful or the harmful categories. In most cases, they just are there. These microorganisms make up the background flora. They're the bacteria that show up when you do standard plate counts or potentially indicator type organisms. These are patiently waiting in the background along with your good and your bad. Now let's talk about how bacteria can grow. I'm hoping most of you recognize this graph. This is the classic microbial curve. In production, when you do a sanitation break, the goal is to get as close to net zero as possible. By doing this, you start your production with the least number of organisms on food contact surfaces. In the lag phase, organism multiplication is slow. 2 becomes 4, 4 becomes 8, etc. If you can keep production in this phase as long as possible, it increases the time available to make safe and quality food. General rule of thumb is 4 hours for lag phase. Next, we enter the log phase. This is where exponential growth is observed, and the number of new cells occurring is proportional to the total population of culture or colony. This is where you grow very rapidly. Once you're in log phase, the microbial load in this process is increasing rapidly, and quality and safety will eventually fail. Stationary phase is the phase when the organism growth slows down. Nutrients are becoming scarce. Waste is starting to build up. The environment they are in is reaching the maximum level of organisms that it can support. Once it reaches that tipping point and the environment can't support any more growth, the culture or colony will enter the death phase. Nutrients are gone, waste has built up, and the environment that was once so glorious and perfect now cannot support growth or survival. So what are the basics for bacterial growth? Almost the same as for our bacteria or our growth and survival: water, food, and optimal temperatures. In the food industry, the basics are very easy to come by for most organisms. In addition to these basics, there are other factors. I have a few listed here. A good example is atmospheric conditions. Some bacteria will grow with an abundance of oxygen. Some organisms, like Clostridium species, will die in the presence of oxygen, and some need a specific amount of oxygen to survive, and some can survive at a variety of oxygen levels: no oxygen, low oxygen, high oxygen. Bacteria are very good at adapting to their environment. They've had millions of years to perfect it, and they still are continuing to adapt today. So let's go back and think about the growth curve and the requirements for optimal bacterial growth. We just discussed this, and think about it in terms of bacterial multiplication. In general, bacteria multiply every 20 minutes. I know, there are organisms that are faster and some that are slower, but in general, under optimal condition, division happens every 20 minutes. So, for example, a bacteria dividing every 20 minutes for 24 hours makes a very big number. Not sure how to say this number, but this is assuming that conditions remain ideal and bacteria are in constant log phase. Now in our example, we say lower the temperature to the lower end of the optimal level, and now division is happening every hour instead of every 20 minutes. Again, assuming the bacteria are in constant log phase, after 24 hours, we are at only 17 million bacteria. I know it's not only, but compared to the unknown number before, 17 million is a significant reduction. This is an illustration of how modifying a growth requirement can control how long bacterial growth and potentially extend your lag phase and possibly production. So one area of focus that controls an important bacterial growth factor is low moisture production. The key to this type of production is keeping the area dry. Well, how do you do that? It's pretty simple. Prevent moisture from being introduced into the area and develop procedures to control moisture if it does enter. Simple, right? Do you know where moisture could be coming from in your process? Well, here are some possible things to consider. Your food product. If you're producing a product with a high moisture content, keeping the area dry is going to be more difficult. Your production or process water. Are you fluming product around the plant? Are you adding product water or liquid ingredient to a step? Are you wet cleaning? I know a lot of dry processes wet clean periodically for a sanitation break. The introduction of that cleaning water is water for bacterial growth until it dries up. So make sure cleaning is effective and you're getting a thorough microbiological clean, and the area is dried up as quickly and thoroughly as possible. Also, if you're cleaning any parts off-site in a wet area, they need to be 100% dry before coming back onto the dry production floor for installation. An area that we see a lot of in microbial issues is the junction of wet product and dry product. So if you have a drying process in your facility, there is an area there where your product is considered wet and then considered dry. Not only is that area sort of wet during production, but when in sanitation mode, wet cleaning meets dry cleaning. And if you're not careful, that water from wet cleaning can be introduced into the parts of the plant where it's supposed to be dry. Keep an eye on humidity. This could be a result of your process, the weather, wet cleaning, or anything that introduces moisture into the environment, and especially the potential for condensation. I've been in processing facilities that were dry, but one piece of equipment was sweating away in the main room along with adjacent piping. That area wasn't really dry, and procedures were needed to control the condensation, as well as having special cleaning needs. Humidity is an easy one to overlook, as some plants, it just comes with the weather. Even if you have good environmental temperature control, if you have a step that involves cooking, steaming, boiling, blanching, anything like that, you could have a humidity issue if it's not controlled. And finally, leaks. They will happen. Have a plan. All right, we having fun yet? So let's start our organism discussion this day with a little microbiology joke. So this one is a modified joke from the Penn State Food Safety blog. Made me laugh. Hopefully it'll make you laugh. So why did the bacteria decide to take up residence in the cow's intestine? Because there was room in it. Get it? The cow's a ruminant. Room in it. All right. I laughed, but I'm assuming you're all laughing. And I'm hoping people like dumb jokes like I do too. All right, back to being mostly serious. Food production comes down to two main things: safety and quality. You can't have safe food that isn't quality, and you can't have quality food that isn't safe to run a successful business. They go hand in hand. We discussed the good, the bad, and the inert earlier. In this section, we'll focus on the bad. There are just over 1,400 recognized human pathogens in the world, with 31 often being associated with food. So we've discussed different types of organisms, how they grow, and generally what they need to grow. Now let's get specific and talk about a few common environmental pathogens you should know. First, we're going to talk about Listeria monocytogenes. Listeria causes an estimated 1,600 illnesses and 260 deaths in a year. The elderly, immunosuppressed, and pregnant women are at the highest risk for severe illnesses. If infected during pregnancy, about 20% of infections will result in miscarriage or stillbirth, with 3% of infants dying after birth. Listeria is a non-spore forming gram-positive rod, and it's a psychrotropic psychrotropic organism. This means that it can grow at refrigerated temperatures. But notice the growth range is wide, going all the way up to 45 degrees Celsius or over 100 degrees Fahrenheit. Optimal pH for growth is 4.4 to 9.4, and the minimum water activity needed for growth is above 0.92. L. monocytogenes illnesses can be tricky to identify as symptoms generally don't appear until a week after exposure, sometimes even a month. Who remembers what they had for lunch yesterday, let alone 4 weeks ago? If you do get sick, whether or not you are at high risk plays a role in the type of symptoms you get. For healthy individuals, you'll get a mild flu-like illness. For high risk individuals, they can be a fever, muscle aches, and possibly GI issues. If illness does progress, they could develop headaches, stiff necks, confusion, convulsions, something very similar to a meningitis-like illness. The infective dose or the number of organisms an individual would have to consume in order to result in illness is just under 1,000 organisms in high-risk individuals. When thinking of pathogens, it's important to know where an organism is found in the environment, as well as how it could enter your facility and develop control measures around that. Listeria is widely distributed in nature at low levels. Because of this, it can enter a facility on almost anything: people, food, objects, you name it. It causes most problems when it's allowed to enter a facility and grow to high levels in the harborage areas. Common harborage sites are floors, drains, coolers and freezers, including in ice buildup, cleaning implements, wash areas that tend to be wet, including in the washers themselves, condensate, remember we talked about that when controlling water, walls and ceilings, especially if they're not cleaned regularly or on your master sanitation schedule, and compressed air. It's very important to test and regular change your filters if you have food contact compressed air. Thorough sanitation to a microbiological level is very important to be sure that these areas don't become possible L. monocytogenes harborage areas. Now let's move to the other important environmental pathogen, or another, I should say, not the other, another important environmental pathogen, Salmonella. Salmonella is one of the most common bacterial gastrointestinal illnesses globally, and it causes an estimated 1.35 million illnesses, estimated 1.35 illnesses, 26,500 hospitalizations, and 420 deaths a year in the US. Getting to know Salmonella a little more, it's like a tongue twister. It's a non-spore forming gram-negative rod. Temperature range for growth is anywhere from 7 degrees to 45 degrees Celsius. And notice the minimum temperature is above recommended refrigeration temperatures. So refrigeration is a common control method, but it will not kill this organism. Optimal pH for growth is 4.4 to 9.5, and the minimum water activity for growth is 0.94. Illnesses caused by Salmonella is relatively quick after exposure, especially when compared to Listeria monocytogenes. Symptoms will usually appear in 12 to 96 hours. They include diarrhea, abdominal pain, chills, fever, vomiting, things that are commonly thought of as GI illness symptoms. Illness will last about 1 to 4 days, and it only takes a small number of organisms to make a person sick, just 15 to 20 in some cases. Salmonella is commonly found in the intestinal tracts of warm and cold-blooded animals, including birds and lizards, if anybody's kid has a lizard at home, and the environment. From this, it enters a food establish on animal food products and contaminated dry foods like peanuts and flour. Salmonella can survive freezing and dry environments. It is a common contamination organism when construction is going on. So if you're going to open up floors, walls, anything like that, be sure to have an implemented plan, including enhanced cleaning and strict traffic controls is required to make sure that this organism does not enter active production areas if it's there. Next on this list, and a newer environmental pathogen, is Coronabacter sakazakii, formerly Enterobacter sakazakii. I use air quotes when saying newer because before the most recent infant formula outbreak, this organism was really only talked about in certain industries, and it's definitely not new. Illness associated with Coronabacter sakazakii is rare. The CDC gets 2 to 4 illnesses reports a year. In 2024, the U.S. added C. sakazakii illness to the list of reportable conditions in infants. This is a positive change that will hopefully help us understand more about the illness's frequency. Those most at risk from serious illness are premature infants, especially infants under 2 months, and people with weakened immune systems and older adults. C. sakazakii is also a spore-forming gram-negative rod. It can grow between 5.5 and 45 degrees Celsius. The minimum temperature is just above the refrigerated temperatures, so refrigeration is an effective control method. Illnesses for C. sakazakii differ depending on who's ill. For infants less than a year old, illness symptoms are fever, poor feeding, excessive crying, low energy, and some may even have seizures. Illnesses in infants less than 2 months old can even get into their bloodstream or cause meningitis. For individuals 65 or older, it can cause bloodstream infections. Thought to be widespread in the environment, testing and isolation of C. sakazakii in foods and on surfaces haven't found its official reservoir yet. But this organism has been isolated from various surfaces in dry food production, in households, assorted food products, and infant formula, just to name a couple of them. Because it's thought to be widespread, the route of entry in the facility can be many different things. Once in a facility, C. sakazakii can also survive dehydration and allows it to persist in environments for long periods of time. And finally, I'll quickly talk about Shiga toxin producing Escherichia coli, or STEX. Although it isn't considered an environmental organism, there have been some high-profile outbreaks in history, and I thought I'd give it just a little bit of an introduction. So there are about an estimated 265,000 STEC illnesses a year in the US, and of those, 36% are caused by O157 serotypes. STECs are a part of the broader E. coli family, which most in that family are harmless. They're gram-negative rods, and they're all gram-negative non-spore-forming rods. With STEC infections, symptoms begin to appear about 2 to 4 days after exposure, and these symptoms will last an average of 5 to 10 days. STEC illnesses are characterized by severe bloody diarrhea and abdominal cramping, but fevers aren't necessarily common. Some people can even develop further into hemolytic uremic syndrome, or HUS, which is a severe complication that involves the kidneys. An infective dose of STEC organisms is also very low at just 5 to 10 cells. All E. coli can be found in the intestinal tract of animals, as well as food or water that is contaminated by feces. Entry into a facility is by contaminated food, unpasteurized dairy products, and contaminated produce. It is important to understand where you are sourcing your ingredients and where your suppliers are to minimize potential contamination. But this is another topic for another presentation. So, and finally, this is a full list of the publication and periodicals I referenced in the organism slide. The websites and the links to those websites were located at the bottom of each page. Now I'm going to pass this over to Joy, and she'll take everyone through chlorine dioxide and considerations for environmental contamination. Thank you.
Thank you, Erin. Thanks for helping us better understand the microorganisms and the challenges that we face every day. Now let's layer in the cleaning challenges that we encounter in food processing. We use a lot of complex, hard to clean, hard to disassemble, hard to reach equipment in a production environment. Then we layer in the types of products produced and the soils, residues that are hard to remove, such as fats, proteins, carbohydrates. And this combination of hard to clean surfaces and hard to clean soils creates that welcoming environment for those microorganisms to hide and thrive. All right, so switching gears, let's talk about a clean break. What is a sanitation clean break? A sanitation clean break is defined as a break in production where all food contact surfaces are documented to have sufficient cleaning and sanitation and are free from contamination. So, if we break down this into the principles from microbiological perspective, a clean break requires effective cleaning or effective removal of the food soil that coats the surface of the production environment or equipment. And once a surface is clean, then it needs to be effectively sanitized to kill any microorganisms on the surface. This is often accomplished using an antimicrobial that is effective against the most challenging microorganism. Effective procedures are also very critical to the cleaning and sanitation process. Different soils and different equipment require different procedures. The frequency at which you implement or establish a clean break is one that would prevent adulteration. So if your conditions are conducive to microbial growth, a higher frequency is needed. If you recall back to one of Erin's slides, under the right condition, they can populate very quickly. And last, and just as important, documentation. Document the cleaning and sanitation process and the results achieved to verify all food contact surfaces are free from contamination. A sanitation clean break can help limit the lot size and the scope of a recall, thereby limiting the risk to your business. One product that will help deliver a clean break, especially suitable for dry production where fewer sanitation tools exist, is chlorine dioxide. So what is chlorine dioxide? Chlorine dioxide is not new. It's been used since the early 1800s. It's a greenish yellow gas with a chlorine-like odor. As fumigators, we like that the gas has warning properties. You can see it and you can smell it. Chlorine dioxide is a strong oxidizer, which gives it its great antimicrobial properties. It is soluble in water, and it remains as a dissolved gas. Most notably, it's used as an antimicrobial agent for drinking water, and for many years it's been used as an oxidizing agent to whiten paper. Chlorine dioxide is a true gas, so it does seek to equilibrate, and it's a very small molecule. It's smaller than most microorganisms that we're targeting, so it can penetrate into those same areas where the bacteria are hiding. Gaseous chlorine dioxide is best designed for those hard to clean equipment and spaces where liquid antimicrobials just can't reach or can't be used. Our gas delivery method allows us to manage the application in a precise and flexible manner. Precision application helps us manage risks like over-application or leaking into non-target areas. We can target specific pieces of equipment by simply tarping them and creating a sealed environment, or we can focus on a product pathway within the equipment itself. We can also scale this treatment to really adapt to any space or size. As a true gas, chlorine dioxide will expand into all of the areas of that treatment zone and destroy the bacteria that harbor in places that cannot physically be reached. So let's review how chlorine dioxide can benefit the food manufacturers like many of you. It has many advantages, but also has some challenges. Let me talk about the advantages first. So we just talked about how chlorine dioxide is a small molecule delivered in a gaseous form and has the advantage of eliminating microorganisms that harbor in those challenging areas that traditional liquid antimicrobials can't reach. We can deliver high dose rates, but because it's a gas, it does not leave behind a residue, and therefore it's safe for treatment on surfaces and does not require a post-treatment rinse. The aeration process sufficiently removes the gas from the treatment area. Treatment parameters can be flexible to limit downtime in production and can be used to respond to a contamination concern or as a preventative approach to ensure clean break between production batches. Routine use of chlorine dioxide provides better contamination control and recall prevention. And chlorine dioxide is a strong oxidizer and a powerful antimicrobial and is considered a sterilant and is effective against the hardest to kill microorganisms. Now, there are some challenges with this technology as well. Chlorine dioxide does not have any cleaning abilities, so it targets the surface or equipment that we're targeting must be pre-cleaned prior to treatment to achieve the best outcome. Chlorine dioxide has some ability to penetrate loosely packed and porous soils, but it really should not be relied upon when surfaces are unclean. This treatment is only approved for equipment and surfaces, so all food, beverages, and ingredients must be removed from the treatment area. Chlorine dioxide cannot be compressed, so we cannot bottle or ship this in a gas form. Therefore, chlorine dioxide is always generated on site with the use of specialized equipment. Because chlorine dioxide is toxic at the dose rates that we're targeting, special licensing and training are required to operate the equipment and perform this treatment. We also need to contain the gas to achieve effective dosage, which means that the treatment area must be sealed off and the area and or facility must be vacated during the treatment process. You'll want to partner with a skilled fumigator that understands gas movement, the best sealing and safety practices. And like other antimicrobials, it cannot prevent recontamination from occurring after completion of a treatment. So it's very important to maintain good sanitation practices after a treatment to keep a clean environment. So now let's talk briefly on the typical escalation to a decontamination service. Most food production plants have routine cleaning and sanitation program that's monitored by their environmental sampling to indicate how effective their program is working. If the microbe results are negative, the routine cleaning and sanitation program is working. If the results are positive, then the cleaning and sanitation program generally gets escalated to something more intense, sometimes more focused, and sometimes this adds labor, more chemistry, sometimes it involves breaking down equipment, taking it apart to get that more thorough clean. When these efforts are not cost effective or when the efforts don't seem to pay off or you're not getting the results that you need, this is a great time to bring in a decontamination service. So when does a decontamination service make sense? I would say we are most frequently contacted when there's a confirmed or suspected contamination, and we can react very quickly to meet those needs. However, there are many opportunities to consider for a more proactive and routine approach. After new construction or renovation, we've heard from Erin that some bacteria can live in walls or floors for years. And after a renovation, these bacteria get moved around and can become a source of contamination. Even with new construction, bacteria can be introduced by construction workers, and starting out with a clean production environment is critical. Decontamination should be considered for decommissioning or recommissioning equipment from storage or movement from another facility. When installing a piece of equipment that hasn't been used for several months, you'll need to spend time cleaning and sanitizing that equipment before bringing it back into production. This is another great time to decontaminate that piece of equipment to make sure it's free of microorganisms. A decontamination treatment can be easily added into planned downtime. This is often when equipment repairs happen or fumigation for a macro pest. We also talk about using this as a clean break between batches to limit risk. Another opportunity might be when a facility is reopening after extended downtime because of seasonal work or repairs, or as a disaster response because of those water pipe leaks or spring floods occur or the roof leaks. These are situations where microbial loads can get really high, as we heard from Erin, and a great time to consider for a full facility decontamination. There's really no limitation on where we can apply the treatment. We just need to be able to contain the gas and achieve proper dosage. Often we are targeting equipment that is hard to clean, hard to break down, hard to reach, or problematic equipment like dryers, spiral freezers, processing and holding tanks, bins, silos, or even those high-risk areas like ready-to-eat rooms, product and system pathways, fluid lines, pneumatic lines, HVAC ductwork. But we can also consider much larger spaces, entire rooms that are filled with equipment, or even the entire facility if needed. Don't overlook those transportation trucks and shipping containers as well. What does decontamination process look like? Well, the first step is always really understanding the specifics of the situation. We have experts at Ecolab that understand your process and challenges and can help identify the best solution. Before we would initiate a chlorine dioxide treatment, we would need to gather information and we understand that every situation is unique. This often requires us to perform a site visit where we would learn from you more about the issues and the specific details of the site and the situation. This is often where we ask and answer a lot of questions from food safety, quality assurance, engineering, and safety. We discuss the issues, what the obstacles that we're dealing with, what timelines are needed, and then we propose an option and we lay out a plan together and how we're going to execute this treatment. We often include multiple viewpoints, process plan, a quality plan, engineering plan, safety plan, just to make sure that everyone's comfortable before we come in on site to perform this treatment. We encourage being proactive and preparing for an event. Doing this pre-work or planning will really save you a lot of time versus if you're in a crisis situation. So let me briefly kind of walk you through the process of what we do when we perform a service. From the previous slide, we used those site visit details and the plan that was laid out. So for this example, let's say that we're doing a large room filled with several pieces of equipment. Our specialist team will come on site, seal off the treatment area, and assess and achieve those proper treatment conditions. And after we're done sealing is typically when we shut down the facility to ensure the safety of all associates. Then next we will run our gas delivery lines and our monitoring lines. And when it's safe to do so, that's when we initiate the gas generation equipment and start introducing chlorine dioxide into that treatment area. Then we will monitor the concentration at multiple different points until we hit our target dosage. We continue monitoring until the end of the treatment is complete. And then once complete, we initiate our aeration process. Our aeration, we aerate until the space is safe to enter. We perform a thorough clearance assessment, ensuring using our low range monitors. Once the facility is cleared and safe to re-enter, we remove our equipment and the sealing materials and we return the facility back to the owner. So if you're thinking of considering a decontamination service, let me leave you with a few things to consider. Think clean, concentration, label, efficacy, affordability, and next steps. So let's start with concentration. So how do you ensure treatment is the best fit for your situation? The first thing that we'll need to understand really is that treatment target area. What needs to be treated? What's that volume of the space? What organisms are we targeting? What dose do we need to be effective for the treatment? How much downtime can be afforded? What flexibility and time do we have? Can we increase concentration to reduce downtime, or can we use a lower concentration and hold for a longer time? Can we increase the relative humidity or temperature to allow flexibility in that dosage? What kind of sensitive materials are in the facility? What other factors can lead to needing more gas? The label. The product label is also very important to consider. While there are many chlorine dioxide products registered with EPA, not all of them carry the same claim set, especially with gas-applied chlorine dioxide. Some products are only registered to be effective against odor-causing microorganisms or as a food contact sanitizer when used in a liquid matrix. But the product that we use is the only one that's registered for use in a gas form as a sterilant. So you should really ask your service provider for a copy of the label early in the process to understand the claims and where and how the product can be used. For efficacy, here's where we want to consider the treatment efficacy. How effectively is it being measured? How effectively is the gas being dispersed? Is the gas being introduced into one location and expected to fill a large room, or will there be multiple points of introduction? Will there be a potential for hotspots or areas of high concentration or weak spots, areas of low or underdosing? Can the concentration be adjusted during the treatment? Where and how many points of monitoring will be used? Monitoring the gas is one of the most important parts of the treatment. This is how dosage is measured, and you'll want a uniform dose rate within the treatment area to avoid that under and over dosing. How will a treatment be verified and documented? You should always look for gas monitoring results to verify proper dosages were achieved. Additionally, we use microbiological indicators as a verification tool of achieving a 6-log reduction against spore-forming bacteria. We also encourage customers to do their own swabbing to ensure that the treatment was effective, leveraging their own tools and knowledge of where the challenges were. Affordability. So let's evaluate the total cost of treatment. What is the difference between the cost of a decontamination service versus manual, more labor-intensive, traditional liquid spray and wipe? In this study, published in the Pharmaceutical Engineering in 2020, the author compared the cost of chlorine dioxide gas decontamination to a traditional liquid disinfection and found that the cost of a gas decontamination was 30% less than traditional spray and wipe approach. Additionally, it decreased the total amount of downtime. Can a decontamination be coordinated during routine scheduled downtime? Holidays and scheduled maintenance are a fantastic opportunity to be proactive in taking advantage of planned downtime. Can the treatment be combined with other activities such as fumigation for macro pests or other preventative maintenance to maximize your downtime? Many times the same people performing a microbial decontamination can also do a fumigation. And I'll briefly address an anticipated question. Chlorine dioxide is not approved as a pesticide fumigant, so we cannot control both macro and micro pests with just this one treatment. Next steps. So once you've committed to a decontamination, how do you get the best outcome? I'd recommend reaching out to Ecolab, and we'll partner with you to establish an open line of communication and to answer any questions you may still have. We'll put a plan in place. We have experienced people helping in many different food processors with their toughest challenges. We want to make sure that everybody understands the plan and how to achieve the best outcome. It's also very important to be ready for the treatment. Make sure that the surfaces are clean so that you get the most effective treatment possible. Secure thorough documentation of the results. This is very important in defending your food safety program. And then last but not least, make sure that you're implementing sanitation best practices for long-term success. Reach out to your sanitation provider if you need any recommendations on how to maintain a clean environment for as long as possible. So before I end, to take questions, let me leave you with an example of how microbial decontamination has helped one of our customers. In this case study, a dry ingredient manufacturer was challenged with presumptive listeria issues when one of their buyers tested a received lot and refused any further shipment until improvements were made and their microbial issues were mitigated. Their buyer recommended them to reach out to Ecolab for support. The three of us, the three parties, worked together to find the best solution for the situation. Our team conducted a root cause analysis of our customer's operation. We found that due to the complexity of the extensive product transfer system spanning the entire facility and much of it being inaccessible manual cleaning alone would not be enough. Per our recommendations, the manufacturer performed a deep clean sanitation, which was followed by gaseous chlorine dioxide decontamination. We achieved a sanitation clean break verified by 6-log reduction of biological spores, and this saved the buyer relationship partnership and renewed the buyer's confidence in the supplier's product quality. This customer now leverages chlorine dioxide as part of their routine cleaning and sanitation program and has scheduled preventative treatments twice a month. Additionally, the customer is going through some infrastructure upgrades and intends to use chlorine dioxide treatment once the renovations are complete. That was my last slide, so I'm going to turn it back over to you, Ashley.
Thank you very much. We've wrapped up our session and now is time for the Q&A portion. Please still feel free to put any questions you have in the chat. We're going to open it up to Erin and Joy. Before I do that, I do want to call out the QR code on your screen on the right side. To learn more and to get in contact with a specialist, scan that QR code and you can fill out the contact form and someone will reach back out to you. For immediate assistance, you can also contact John Miller. His email is on the screen, but it's john.miller@ecolab.com. I don't see any questions in the chat, but I do have some for you. So Joy or Erin, either of you feel free to jump in. First of all, can chlorine dioxide be used in an organic production facility?
I'll take that, Erin. Yes, great question. Chlorine dioxide can be used in organic processing facilities. According to the USDA National Organic Program list of cleared substances allowed as sanitizing on non-porous surfaces. I think it's 7 CFR 205. However, with any material used in organic production, it should be included in your organic system plan, the OSP, and the plan should be reviewed with your organic certifier.
Okay, excellent. I do see questions coming in, so I'm going to read some from the chat. Is there a reason we couldn't do this in food service/retail?
No, there's not a reason we couldn't. Generally speaking, it's a smaller space, right? We just have not. I would love to learn more about your opportunity. There's no reason we couldn't do it there.
Great. Next question. What is the main advantages and disadvantages of chlorine dioxide versus ozone treatment?
Good question. We don't see ozone very often. It is out there. Ozone is also another strong oxidizer, a little bit stronger than chlorine dioxide actually. It degrades very quickly though when it comes in contact with anything organic. Chlorine dioxide does too, but ozone is much more reactive. So it's harder to generate ozone as well. You have to use pretty specialized equipment to generate ozone. In my experience, it's harder to fill spaces, the large spaces that are needed for a food and beverage production environment to get effective dosages of ozone versus chlorine dioxide. Chlorine dioxide lasts longer in those scenarios and generally is more effective, and that's coming out of published papers. So that's my experience.
Okay, excellent. Next question. Outside of dry facilities, what are some common use cases for gaseous chlorine dioxide?
Another great question. We did talk a lot about this being used in dry facilities, and that's, the dry industry really just doesn't have a whole lot of tools. So this is a great fit for the dry industry, but we also use it in many other traditionally wet cleaned facilities. It's similar equipment. We've done it with spiral freezers, piping systems, aging rooms, CIP systems, aseptic filling lines. So I would say there's really no limitation to where we can apply this. We just need to be able to maintain the gas to achieve that effective dose.
Okay. What is the typical response time for an emergent microbial issue?
Well, it all depends, right? we, uh, we typically respond to any customer inquiry within a day. Um, and we can be on site very quickly soon thereafter. We can mobilize a team as needed. We are strategically located around the United States. We just have to be able to get our equipment on site. And so it's really just that transportation time that's needed. But in general, we can respond to any emergent issue that might happen. So.
Okay. Excellent. And how long after the chlorine dioxide service can you enter the facility and resume production?
Another great question. And that, I would say, as soon as the treatment is aerated and we can confirm that the gas is out of the facility by monitoring, then we, technically speaking, you can start up production right away. There's no requirement to rinse post-treatment. So as soon as the facility is able, they can get right back into production. I would say after a treatment is, if you're going to be doing any swabbing, we would recommend that you swab after a treatment and before production resumes, just to get that post-treatment assessment.
Okay. You kind of touched on this a little bit, but one question in the chat is, what are the latest developments in chlorine dioxide treatment to reduce or optimize preparation time for gassing and aeration?
I would say the equipment has definitely improved. We have much more mobile equipment right now. We can kind of target lots of different areas, spots within the facility or within a production line. So it's just gotten more precise, I would say. But in general, chlorine dioxide is the same, chlorine dioxide.
Okay. Another question from the chat. You've mentioned that this works well in hard to clean areas, but that said, it was also stated that most organic material must be removed for this to be effective. Do we have anything you can use in tandem to ensure organic material is removed from hard to reach areas before the gas treatment?
Yeah, and that is a trick. And I would say that would be a great opportunity to partner with your sanitation provider to find a good way to clean your facility before the treatment. It is, in some cases, very hard to do, whether that be a liquid clean, whether that be a dry clean. So I think it's going to have to depend on the situation and the scenario that you're challenged with. But reach out to your sanitation provider. They can help provide some good cleaning practices for those specific areas.
So can I just add one more thing to that as well? When you're talking about getting into hard to reach areas, when we talk about sanitation beforehand, breaking down your equipment, as far as you can, and making sure that you're cleaning in those areas is also extremely helpful. If you gas when the equipment's broken down, obviously it's much easier for that gas to penetrate hard to reach areas or areas that might normally be hidden.
Okay. Would you suggest ATP swabbing prior to treatment?
Prior to evaluate the cleanliness of the surface, I personally recommend bacterial testing. ATP will help with kind of the soil load. It does an okay job looking at bacteria, but it can also kind of mess up some of the parameters of ATP. Maybe Erin, you can help me with this one.
Yeah, so
there.
Yeah, I agree. Microbiological swabbing would be good, doing kind of a beforehand and then an after so that you're aware, so can you kind of know where you started and where you're going to end up. But if you're doing thorough cleaning, especially if you're breaking down equipment and exposing areas that haven't been cleaned in a while, ATP may be able to help you determine, are you clean enough? Is that surface truly clean? So, it's definitely a tool. But again, to Joy's point, it's a verification of clean, not necessarily a verification of microbial cleanliness.
Is treatment by Ecolab only an option in the US or in Canada as well?
Another very good question. It is only available, it's only approved, I should say, by the EPA in the United States. The label does not have approval in Canada.
Okay.
Sorry, Canadian.
I think we have one more question in the chat. What is the best bioindicator you recommend to validate effective gas treatment and why?
We use Geobacillus stereothermophilus. That just tends to be what the industry uses the most for validating sterilization. We have used Bacillus atrophius as well, also another good one. So either of those two, you need a special wrapper around those. There are different ways that those spores are manufactured. You need to have a Tyvek wrap around them because that allows the gas to get into the spore strip, but it doesn't allow the bacteria to get out. So you just want to make sure that you're using the right production method for those spore strips. And then I would recommend Geobacillus stereothermophilus or Bacillus atrophius.
Great.
And just to clarify, Joy, when you guys do the treatments, you do supply the strips as well, correct?
Yes, we actually do those strips, and we can help. We do those strips every time. Thank you, Erin. And we can help with the customer set up that step, that verification step.
Excellent. I don't see any other questions in the chat, and we have 2 minutes left, so I think it's time to wrap it up. Thank you so much, Erin and Joy. You provided us with so much information. Again, a reminder to everyone, this was recorded, so we will be sending this out. And please feel free to contact us for more information. Again, that'll all be sent to you. Thank you very much.
Thank you, everybody.